In this video, we'll be looking at the terminology of agonists and antagonists as drug activities. Here are the learning outcomes that we'll be looking at in this particular video. So we're going to explore how drugs and targets interact with each other. Now, we'll start by looking at that this is a dynamic process. So therefore, drugs and receptors are in equilibrium with the drug receptor complex. And then that drug receptor complex can be activated into a more active form. Now, the first point is that often this is described as a lock and key scenario where you need to have the right key to fit in with the right lock. And for a molecule, what defines the best fit is a number of different properties. So the first point is that they're sterically compatible with each other. So the shape is important for the ligand or the drug to bind to its receptor. The other point is there are chemical recognition sites as well. So if we're looking at hydrogen bonding, they need to be compatible with each other. We might be looking at pi interactions with aromatic groups. We might be looking at van der Waals forces, or we might be looking at ionic changes. Now, all of these things have to work together in the recognition site with that drug for the drug to bind. Now, the binding or the strength of that binding is dependent on the complementarity of the receptor to the drug or to the ligand. And the second point is that there is often what is seen as an induced fit. So there is a conformational change in the receptor after it binds. So there is a movement. It's not a static thing. This is actually a highly mobile protein. So not only is there an induced fit at the recognition site, but you can also get new sites being opened up. So there could be a new protein binding site that is caused by this drug binding to the receptor in the first place. And there are a couple of terms that we'll explore at this point as well. So the first term is specificity. So specificity can be seen as a reflection of the number of different drug targets or targets a drug recognizes, but also their location. So the receptor can actually be that is being recognized can actually be found in a number of different tissues, and therefore has the specificity of its effect is going to be variable depending on where those receptors are found and which receptors it can bind to. So a highly specific drug will bind to a limited number of targets in a limited number of locations. So that gives you a very specific effect. A less specific drug is going to bind to more different types of receptors, and they might be found in more different locations or a combination thereof. So that's the term specificity. Next term, selectivity, is more concentration dependent, and it reflects the concentration dependency of the binding. So it might have not perfect specificity, so it might bind to a couple of different receptor types. But if it does so at a different concentration, you might be able to get it to bind at a lower concentration to the target that you're interested in and not to the other targets which you might need a higher concentration. And this is the case that you'll find with most drugs, is that they're not purely specific to a single target. They're more likely to hit a number of different targets. But what you may find is that there is more selectivity in the process because they're concentration dependent. And ideally what you would find in a clinical scenario is that with the correct dose or the correct concentration, you can get a selective effect where you'll get your target effect without your off-target effects. Okay, so what we're going to look at now is the kinetics and the relationship between a drug and its concentration or dose and its response. Now, usually dose is used when you're talking about tissues and organs and bodies that you're introducing this drug to. If you're talking about a more in vitro situation, like a test tube, then what you'd be looking at is more concentration, because the concentration at the receptor side is going to be more well characterized. And so if we plot what happens to the output if we're monitoring a particular cellular function or a cellular response, that usually goes on the y-axis, and the dose usually goes on the x-axis. So if we just use normal dose, just straight linear, what you'll see is this kind of asymptotic kind of behavior as it reaches a saturation point where the cell and its setup, its infrastructure, and the number of receptors and the links to the output are all fixed. You get a point where you can't do any more. The machinery of the cell has reached its maximum limit. And so that's what we're seeing here. Now, if you do the same kind of thing with a log dose, you'll see saturation occurring as well. But you'll also see down the bottom that there's a point when the concentration gets to a high enough concentration when you're actually going to start to see that response starting to appear. But below that, nothing's really happening. There's just not enough drug available around to activate the receptors that are present. So there's your saturation point, and the other point is your threshold dose. So this is a dose that you need to reach to actually get to an effect or see an effect. Okay, there are two ways of looking at these responses. So the first way I'll talk about is dose concentration, or dose or concentration, and we'll stay with the logarithmic one because it's just easier to see what's going on. And we'll map that up against occupancy. So this is the amount of drug bound to the receptor. Now, please note that occupancy doesn't always lead to an effect. And hopefully at the end of this video, this will kind of gel. This will make sense why that's possible. So here we've got, as we increase the concentration, we get to a point where we're getting drug occupying the receptor up to the point when all the receptors are bound to drug. So we've reached 100% occupancy. Now, there are a number of points on this curve that are of interest. The first point is a saturation point. So this is the Bmax, or the binding maximum. So this is the maximum amount of drug bound in this system. You can't get any more bound to that particular receptor because they're all the binding sites are full. The next point is the Kd. Now, this is a dissociation constant. And this is a concentration at which half the receptor population is occupied. So there's your halfway point of occupancy, and here is the concentration, which is the Kd for that to occur. Now, this is actually based on the on and the off rate. It's a kind of a ratio between the two. And so if we have a look at this equation up the top here, so we have the drug and the receptor being as separate entities, and then the drug bound to the receptor. Now, the Ka, which is the on rate, is the kind of affinity or the association of the drug to the receptor. And this seems to be fairly constant. So it's almost like a Brownian motion. It's an accidental occurrence. Now, what's different, though, is because the recognition site recognizes the drug and is complementary to the drug, it will actually have a stronger binding strength between the drug and the receptor. So its off rate is going to be slower than its on rate. And so therefore, the Kd is affected more by this dissociation rate. And so the slower that dissociation rate, i.e. the smaller that is, the higher the affinity of the drug for the receptor and the receptor for the drug. So if we're comparing drugs to each other, what we find is that the lower affinity drugs actually have a Kd which is higher. So this occurs at higher concentrations. So what this means is that to get a low affinity drug to bind to a receptor, I need more of it. So I need lots more of the drug to actually stay on the receptor for longer periods of time until I reach a saturation point. Whereas a high affinity drug will bind even minuscule amounts of drug and therefore will be bound to saturation with lower concentrations. So this is what we're looking at with the KD. We're talking about affinity, and the lower that concentration at which that occurs, the higher the drug's affinity for the receptor. So another way of looking at the relationships between the concentration or the dose of the drug is to look at its response and that relationship there. So the curve looks very similar, but the kinetics that we looked at previously only went so far. So they looked at the drug and the receptor binding to each other, but that's as far as it went. The next step is to consider the kinetics of the drug and the receptor and the conformational change to the active form of the drug receptor. So this is where the secondary messenger pathways get activated, and it needs to be in this form for that to actually occur. So the relationship has similar points, it has similar features. They have different names when we're looking at a concentration response curve. So the first point is the Emax. So this is a maximum response from the drug in the system. So it's the maximum that can be hit. And that occurs at this kind of concentration here. Now, the next point on the curve, again, is that 50% response mark. But instead of being a KD, what we're looking at here is the effective concentration at which you achieve 50% of the maximum response, or it could be an effective dose. So it depends on whether you're looking at tissues and bodies, or if you're looking at more of a test tube scenario. And we can use these features again to compare drugs to each other. So if we're looking at the term potency, like the KD, what we're looking at is that if it is small or low, if the effective concentration at 50% is small, then it's a more potent drug than a drug which has a higher EC50. So if we have a look at this one, the system is responding to the drug at lower concentrations. So therefore, I need less drug to get the response. Whereas for this blue curve, this which is less potent, I need more drug to get the maximum response. So this drug is more potent, and this drug is less potent. The other way of looking at this system, and the other comparison we can do, is we can look at the intrinsic efficacy of the drug and the receptor. So if the curve is lower, if it doesn't reach 100%, then that means the efficacy or the intrinsic efficacy is lower. And we'll look at how that influences the terminology that we talk about when we're talking about drug reactions and drug activity. You may also see the term efficacy, but this is tissue dependent. So intrinsic efficacy is useful for comparing the drug activity at different tissues, but efficacy is useful for if you're just using the same tissue and using different drugs. So there's a slight variation in the difference between efficacy and intrinsic efficacy. Right, now let's have a look at where this efficacy comes into terminology. So again, we're looking at drug receptor, drug bound to receptor, and then the activity. So if we have a look at our first one, so this is our first curve. So this is a full agonist. So it's reaching basically your 100% response rate. The next terminology that we'll see is a partial agonist. So regardless of how much drug I put in, you can see I've reached saturation and I cannot get to the maximum response. So this is where the efficacy or the intrinsic efficacy of the partial agonist is less than the full agonist. So we can compare drug A with drug B by looking at their efficacy. And you can see that the partial agonist has lower efficacy compared to the full agonist. Sometimes you might see the full agonist saying that this is basically an efficacy of 1 and the partial agonist has an efficacy less than 1. The next drug class that we'll look at is the antagonist. So the antagonist binds, so we get this far, but it doesn't take it any further. So the antagonist has actually bound to the receptor, but doesn't make any conformational change. It's basically occupying the binding site, so therefore no other ligands can react with it. The final one that we're going to look at is an inverse agonist. So this is a little bit confusing. We've got agonists at the top of this and we've got agonists at the bottom of it. So the way to think about this one is if you see the term agonist, what you're seeing is a drug that can cause a conformational change in the receptor. Now a full agonist and a partial agonist are going to make that into its more active form, whereas an inverse agonist is going to make it into an inactive form. And to kind of help unpack this a little bit further, what I'd like you to do is have a look over here and you'll notice that this is not at 0. Now, there are some tissue systems that the receptor in that tissue is actually active without any ligand being available. It just randomly turns on. And so therefore you always end up with a little bit of signal in the baseline. So the baseline is non-zero. This is referred to as constitutive activity and is required to detect the presence of an inverse agonist. So what the inverse agonist is doing is it's stabilizing the receptor in its inactive form. And so therefore the agonist is actually, or the inverse agonist is causing a conformational change, but it's to an inactive state, whereas the partial agonist and the full agonist are converting that protein into and stabilizing it in a more active state. The other thing that we're going to look at here is there are different types of antagonists, just as there are different types of enzyme inhibitors, and the terminology is very similar. So if we're looking at competitive antagonists, so these are binding to the same site where we have one orthostatic binding site and both the antagonist and the endogenous ligand can both bind to the same site. And this is an example of a reversible one, so the antagonist can bind and it can dissociate. You can also have irreversible competitive antagonists, again binding to that orthosteric binding site. The drug binds and leaves a little bit of itself behind covalently and therefore antagonizes the receptor on a more permanent basis. And the final one we'll look at is the non-competitive antagonist, where you have an orthosteric binding site and you also have allosteric binding sites. So the orthosteric site is where the endogenous ligand binds. The allosteric is basically where the endogenous ligand doesn't bind. So it's somewhere else, usually on that protein. And so both of those can interact with the protein independently from each other. And this provides us with a non-competitive antagonist. Okay, so we're back to the learning outcomes. So you should be able to explain how drugs interact with proteins. So the steric and the ionic, those kind of interactions, the conformational change, those kind of things are things that you should be able to describe or explain. You should be able to look at the term affinity and explain what that actually means with respect to drug and receptor binding. Should be able to differentiate the terms specificity and selectivity, one being more concentration dependent. The other one is to differentiate intrinsic efficacy and what that tells us about the drug and where that ties in with full agonists and partial agonists and antagonists, and what potency can tell us about how drugs are different from each other. And finally, compare different types of drug activity. So these are the partial agonists and the agonists and the competitive antagonists and the non-competitive antagonist.